In this segment, I'm going to talk some more about resources for pharmacogenomics. So I divided the resources for pharmacogenomics up into three groups. One segment for clinically relevant, more clinically relevant type information. One segment for more basic drug and genomics information. And one segment for PharmGKB. So in this segment, I'm going to talk about resources for drug information and genomics. Specifically, Drug Bank, the UCSC Genome Browser, and some resources at NCBI. So Drug Bank is a drug information resource that's used by PharmGKB, which you'll hear about in another segment, pretty extensively. It has detailed drug information about a huge number of drugs. They have 8,312 drug entries. And you can see the breakdown is a couple thousand of those are FDA-approved small molecule drugs. There's a couple hundred biotech drugs, which means they're like proteins or peptides used therapeutically. There are even 93 nutraceuticals, and a whole lot of experimental drugs in there as well. And each one of these drug entries, can have over 200 different fields of information available. So I'm going to show you a screenshot of the homepage. The homepage always has a drug of the day down below. So if you're interested in browsing more information, you can just click there. But if you want to search for a specific drug, there's a search box up at the top. So I'm going to pretend like I enter clopidogrel in here. And this is the results page for this particular drug. It has known PGX associations. There's only one search return in this particular case, but just as a heads up, oftentimes there are many, many returns for a particular drug. So here we have clopidogrel. I'm going to go ahead and click on that entry because I know that's what I want. And it takes me to the full drug entry for clopidogrel. There are really broad general descriptions, the structure of the drug, synonyms for the drug at the top. But then as you scroll down, the information can get more and more specific. There are tons of fields. You're going to scroll for quite a while down this particular page. Many fields about the forms the drug is available in, companies who manufacture the drug, the route of the medication, oral or intravenous or what have you. They have information about the prescription generics and brands at the top. They have a lot about the the pharmacology of the drug, both the pharmacokinetics and the pharmacodynamics. So it has a lot about mechanism of action, a lot about how this drug is absorbed and metabolized. An important section on Drug Bank for pharmacogenomics is that they do contain a section on SNPs that have been associated with drug response or metabolism, and also adverse reactions. They have a section on drug targets as well. So each gene that has been identified as a drug target, they give you references that you can look to for more information, and a link to UniProt to learn more about the particular gene. I have a section on metabolizing enzymes in this case. They have CYP3A4, but if you scroll down, you'll see the same old CYP2C19 that has been mentioned with clopidogrel in many of these segments. A great thing about Drug Bank is that they have a downloads button at the top of every page. That bar along the top has a downloads button. If you click it, it takes you to a page where you can download all the drugs in Drug Bank, and then you can manipulate it to your heart's content, and you can group drugs based on any of the different fields that are available. So it's kind of a good mining resource if you're looking to do bioinformatics work with drugs. Okay, I'm going to switch gears away from drugs and go to genomics. Talk about the University of California, Santa Cruz Genome Browser. The genomics group at Santa Cruz has a suite of tools that they make available, and one of which, the main tool really, is this browser called Golden Path. The current assembly that they use is HG38, but you can go back and look at other assemblies as well if you want to look at a previous genome assembly. And you're able to search, basically search the genome by position using either chromosomal coordinate, or you can give it a range, like a gene range. You can put a gene name in, and you can also search by different track types, and I'm going to explain what the tracks are. So this is the screenshot of the home page for UCSC Genome Bioinformatics. So if you type that URL in, and you want to click on this little button on the top left, that will take you to the genome browser. And the browser is a really complicated display that looks kind of like this. I hope you can zoom in on this maybe on the slide if you can't see, or just go to the website and follow along. So every entry along here is a different track. And you can see different genes. If you zoom in, you can see we're looking at CYP2D6 here. You can see where the introns and the exons are on that gene. You can see where variants have been detected on a particular gene. Depending on what track you're on, you can see clinically relevant information. There are ways to modify the tracks that you show. And I'm going to show you a little bit about that. You can turn on and off different tracks and zoom in on the places that you really want to examine on the genome. So up here, it's kind of showing you where on the chromosome you are, this little red box. Okay, so I kind of zoomed in on that top part right here. These are the controls to move along the genome, so I'm going to zoom in on that, make that bigger. So you can see you can move up and down the piece of DNA with arrows. You can zoom in. You can zoom all the way down to the base level. So if you want to see the actual ATGC code, you can see that for sections of it. If you click zooming down to the base, you can zoom out again. And you can enter a particular position and go straight to that position by typing it in here. I typed in CYP2D6 to start off with, so we're looking at a region of the chromosome. So I zoomed in a little bit further on this particular, on the grid, so that you can kind of see some of these tracks down the side. You can see where there are mRNAs, spliced ESTs. There's different, we're looking at comparison between the human genome and different model organisms here in this section of the screen. We're looking for common SNPs. So these are SNPs from dbSNP, which is an NCBI resource that I'm going to tell you about a little bit later. So you can see where there are actual known variation on this region of the particular gene. So the entire genome browser section, this picture section, is at the top of the page. And if you scroll down that page below this view of it, you're going to have a section that looks like this. And this is listing all the tracks. Each one of these items, kind of check boxes, show up as a track along here. So you can turn these on, you can turn them off, you can hide them. You do have the ability to customize. You can upload custom tracks if you'd like to. I'm not going to go into that today. There are procedures for doing that found on the website. It can be kind of complicated, and I'm not going to take the time to go into it today. But people do, if you have your own experimental data and you want to map that onto the genome and see it as a track, you can definitely do that as well. This resource is really great if you want to find, for pharmacogenomics, if you want to find known variation across that gene, if you want to be able to do comparisons to other organisms. So that's, I think, you can also do BLATs. You can BLAT sequences that you have to the human genome through this particular tool. And so those are the kind of common uses for the UCSC browser. The last thing I'm going to talk about is NCBI. NCBI has a huge suite of tools and databases available. I've already talked about ClinVar and something called the GTR, which is the Genetic Testing Registry, in a different segment. Of course, PubMed is part of NCBI. And they have a lot of genetics and genomics tools and databases, as well as those regarding RNA, proteins, bioassays, all kinds of things. So I'm going to show you what the home page looks like here. This is welcome to NCBI. So there are so many options to choose from. You can enter a gene or a drug name in this search box at the top, and it will search across all databases and bring back a search return for you across all of them. It might be from a DNA resource, it might be from an RNA resource, it might be from a protein resource, and if they all have a hit for that particular word that you typed in, it'll return all of them to you, which is really nice. But I'm only going to talk about a couple specific tools or databases or resources in NCBI, and that's dbSNP and RefSeq. So I'm going to start with dbSNP. dbSNP is a database for SNP information, but it's also more than that. It's just any submitted variation to the resource. So any variation that's submitted, it gives, dbSNP gives a unique ID to it. It's called an RSID. And so every number starts with RS, and then it's a string of numbers. So it's difficult to remember that number. It's kind of meaningless on its own, but the value in it is that it's a unique ID for variation at a given position in the genome. When you're submitting to dbSNP, you can submit flanking sequence on each side of your variant that you have found, about 20, 25 bases upstream and downstream, and they will blot that against the genome and align it for you, and then give that position that unique ID. Or just recently, they started to be able to accept VCF formatted files with the position on a reference sequence already. You can give them a position on a reference sequence and submit that directly to them as well. They used to only take the flanking information. Even though the name is dbSNP, they take more variation than just SNPs, or single nucleotide polymorphisms. They take information on indels, some repeats, and though SNP implies a particular frequency, there's no frequency requirement for depositing to dbSNP either. So it could be extremely rare or extremely common. dbSNP is just a name. So this is the home page. It's a great resource for uniquely identifying positions for a variant, but there are some drawbacks to it as well. For example, if you don't already know the RSID for the variant of interest, it's kind of difficult to find that. You can enter a gene name in the search box, such as TPMT. I know the variant that I'm interested in is on this particular gene, but I don't know that random string of numbers that is the RSID. So if I type in TPMT, I get 8,365 hits, which is too many to search through manually. And they do give you some options for filtering on the left side of the screen to help narrow your list down. So if I restrict the search to Homo sapiens and maybe variants that are classified in ClinVar as pathogenic, then I get 13 results, which is much more manageable, and you can look from there. But it is, dbSNP is more useful as a resource if you already know the dbSNP, the RSID of the variant that you're looking for, and then you just want to find more information about it. So we're going to pick a SNP, and I'm going to show you how it's useful once you already find your variant of interest. So if I follow the top variant, or one of those variants on the previous page, I'm going to get information about the possible nucleotides at that position. So for example, in this case, we're looking at an A or a G. I get all of the HGBS names for mappings on chromosomal sequence, gene sequence, mRNA, protein. I get the alleles, the amino acid changes are described for me, the allele changes and amino acid changes for this particular variation, and with positions and mapping to reference sequences. If you scroll down that page, sometimes there is allele and genotype frequency information from submitters. So this is a list of all the population groups that have had information for this particular variant submitted for them, and then the frequency of the genotypes and of the alleles. That can be very valuable if you're looking to understand the frequency of a particular SNP or variant in a given population. But you kind of are at the mercy of what's been submitted to dbSNP. This is not a comprehensive frequency list, and it will vary highly from SNP to SNP. So there are some variants entries in dbSNP where there's no frequency information at all. But for example, a SNP that's very well studied has it nicely laid out for you, and you can compare across populations. So the other NCBI resource I wanted to talk about is RefSeq. It's a reference sequence database. Each sequence is in only once, it's non-redundant. It contains genomes, transcripts, and protein sequences. As the sequence gets updated, so with every version of the genome that comes out, the reference sequence can be updated. The sequence gets refined. And instead of depositing a new sequence into RefSeq, what happens is they version it. So you have version control. So the first sequence that's deposited is a 0.1, sequence 1, 2, 3, 4, 0.1, and then the next one is 0.2 and so forth. So just be aware that with RefSeq sequences, the main sequence name is not enough. You have to have the version number after that decimal point. Is very important because the actual sequence can change between versions. It's meant to be a stable reference for genome annotation and for using HGBS nomenclature. So being able to map a position on a reference sequence, that's what these are intended for. This is the home page for RefSeq. These sequences are very important for mapping, as I just said. I'm going to search TPMT, put TPMT in the search box at the top. I come back with a lot of reference sequences, but if I'm only interested, you can use the filters on the left. I'm interested in human TPMT. I can see it right there. We've got some mRNA. And if I click that link, it's going to take me to a page where I actually have the entire sequence laid out for me. Is useful in certain situations, not in others. At the top of this file, though, is some information that can be really useful. It's annotated. So that sequence is annotated, and all that information is found on that page in RefSeq. So for example, in this case, it's going to tell me where the exons are. I can see all the different exons. I know the positions, the base positions for each exon, where the coding sequences are. I have cross-references to HG and C sometimes. It's not listed on this particular page, but if you scroll down, you would see a cross-reference to the gene. Oh, no, it is right here. The cross-reference to the gene in HG and C, which is the Human Genome Nomenclature Committee, so the official symbol for that particular gene. If I follow that link, then I'll see other synonyms for that gene so I can understand, map across different databases so that I know the different names for that particular gene. So RefSeq is useful in combination with dbSNP in order to map variation, in this case pharmacogenomic variation, that is responsible for a particular phenotype in people. So a lot of times in the literature that comes out, they will reference a variant based either with the dbSNP ID, so now you know how to find more information about that particular variant given that dbSNP ID in the paper, or sometimes they'll just give you HGVS and a position, and the HGVS is typically on a RefSeq, and so that's what the RefSeq NCBI resource is for. So I hope that gives you a kind of an overview of a few resources that are available for learning more about the genomic information or the drug information in pharmacogenomic associations.